Hi, my name is Ali Sajid Imami. I'm a Fulbright scholar and master's student at the University of Toledo and a part of the Cognitive Disorders Research Lab. Today, I will be talking about balanced output of RNA editing, or BORD, an RNA editing detection pipeline we have developed at our lab. RNA editing is a rare form of post-transcriptional modification. RNA editing is seen most commonly in two forms, the A to I editing and the C to U editing. These effectively replace an A with a G and a C with a T, respectively. So why care about RNA editing? RNA editing is an important mechanism for tissue-specific protein expression, like generating ApoB48 and ApoB100 from the same sequence. It has also been associated with certain pathological processes like autism spectrum disorders and schizophrenia. A recent paper characterized SARS-CoV-2 and its relationship with host-dependent RNA editing. Above all, it's a fascinating process and a great addition to the omics. However, characterizing the editome is a difficult process by its very nature. Editing is a rare event. The biochemical methods are slow and require sites to be known, and on sequencing, editing is indistinguishable from single nucleotide variants. The approach to edit calling can be broadly summarized as follows. You align the transcriptome to the reference genome and then call variants. You then filter out lower quality reads and lower quality mismatches, for example the one that read edges. Then you remove the known SNPs and do some more filtering. At the end, you annotate the results and see which genes and sites were edited. There are several tools available that use sophisticated filtering and advanced analyses to find potential editing sites. Many labs have written their own custom pipelines as well. However, there are still a few problems with the existing tools. They are CLI applications and require a level of tech savvy to use correctly. They also rely on several specific annotation files that have a custom format or that may be harder to generate. Finally, the memory and processing constraints can sometimes be prohibitive. When we ran into this problem, we decided to not only reinvent the wheel, but create a better and more efficient one. Balanced output of RNA editing is a cloud-native pipeline that we have developed for high-performance editome characterization. Our pipeline is designed to require minimal user input and requires no complex annotation files to be generated at the user's end. BORD is powered by Amazon Web Services. This is a typical workflow for the end user. The only input required are FASTQ files. The user uploads the sequencing files to an AWS S3 bucket and then starts a BORD CloudFormation stack. This then creates a compute cluster, downloads and processes data, and then saves the result in another S3 bucket with no user intervention required. Behind the scenes, we rely on containerization and a microservice-based architecture. Any annotation or reference files needed are curated on our end and made available to the compute cluster. The results are also stored in the cloud, which makes it easier to integrate into downstream analysis workflows. This figure summarizes our technical workflow, which is not much different from the standard approach. We start with FASTQ files. We then proceed with alignment and variant discovery using standard pipelines. We then exclude known SNPs using existing databases like 1000 Genomes Project and dbSNP. This is then followed by several stringent filters so that the resulting list of variants is most likely to contain edits. This is then output as VCF files and summary tables. BORD is currently in a closed beta within our lab. BORD will be available in three forms: a CloudFormation stack for AWS, a Docker container with batteries included, and a CLI, which is very do-it-yourself as regards to the associated annotation files and software dependencies. Going forward, With the first release out, we would like to do a lot with Bohr. This includes, but is not limited to, developing intuitive visualization techniques for editome data and a public server where people can use Bohr readily. Especially, we would like to integrate differential editing analysis into the pipeline. In the end, I would like to acknowledge Dr. McCallum Smith and Dr. Shukla, who are my advisors, and my fellow graduate students in my lab for all their support. I would also like to thank the University of Toledo, the Fulbright Commission, and Eastern Medical University for bringing me to this stage. Thank you.